Hello students, today we will learn about germplasm conservation on cryopreservation. How you can preserve the seeds. Germplasm broadly refers to the hereditary material transmitted to the offspring through germ cells. That is total content of genes. The germplasm provides the raw material for the breeder to develop various crops. Thus, conservation of germplasm assumes significance in all breeding programs. As the primitive man learned about the utility of plants for food and shelter, he cultivated the habit of saving selected seeds or vegetative propagules from one season to the next one. In other words, this may be regarded as primitive but conventional germplasm preservation and management, which is highly valuable in breeding programs. The very objective of germplasm conservation is to preserve the genetic diversity of a particular plant or genetic stock for its use at any time in future. In recent years, many new plant species with desired and improved characteristics have started replacing the primitive and conventionally used agricultural plants. It is it is important to conserve the endangered plants or else some of the valuable genetic traits present in the primitive plants may be lost. A global body, namely International Board of Plant Genetic Resource, in short IBPGR, has been established for germplasm conservation. Its main objective is to provide necessary support for collection, conservation, and utilization of plant genetic resources throughout the world. There are two approaches for germplasm conservation of plant genetic material. First is in situ conservation, second is ex situ conservation. First of all, we will learn about in situ conservation. In situ, what do you mean by in situ conservation? The conservation of germplasm in their natural environment by establishing biosphere reserves is regarded as in situ conservation. This approach is particularly useful for preservation of land plants in a near natural habitat along with several wild relatives with genetic diversity. The in situ conservation is considered as a high priority germplasm preservation program. The major limitations of in situ conservations are first is the risk of losing germplasm due to environmental hazards. Second is the cost of maintenance of a large number of genotypes is very high. Now let's know about ex situ conservation. Ex situ conservation is the cheap method for the preservation of germplasm obtained from cultivated and wild plant materials. The genetic materials in the form of seeds or from in vitro cultures can be preserved in gene banks for long-term storage under suitable conditions. For successful establishment of gene banks, adequate knowledge of genetic structure of plant populations and techniques involved in sampling, regeneration, maintenance of gene pools, etc. are essential. Now let's know about germplasm conservation in the form of seeds. Usually seeds are the most common and convenient materials in conservation of plant germplasm. This is because many plants are propagated through the seeds and seeds occupy very small space. Further seeds can be easily transported to various places. There are, however, certain limitations in the conservation of seeds. Variability of seeds is reduced or lost with passage of time. Seeds are susceptible to insect or pathogen attack, often leading to their destruction. This approach is exclusively confined to seed propagating plants, and therefore it is of no use for vegetative propagated plants, that means potato, like these plants. It is difficult to maintain clones through seed conservation. Certain seeds are heterogeneous and therefore are not suitable for the true genotypic maintenance. Now let's know about in vitro methods for germplasm conservation. In vitro methods for germplasm conservation. In vitro methods employing shoots, meristems and embryos are ideally suited for the conservation of germ of vegetative propagated plants. The plants with recalcitrate seeds and genetically engineered materials can also be preserved by this in vitro approach. Let's know about the advantages of in vitro method. First point is large quantities of materials can be preserved in small space. Second point is the germplasm preserved can be maintained in an environment free from pathogens. Third point, it can be protected against the natural hazards. Fourth point, from the germplasm stock, large number of plants can be obtained whenever needed. Next point is obstacles for their transport through national and international borders are minimal. Next point is there are mainly three approaches for the in vitro conservation. First is cryopreservation, that is freeze preservation. Second is cold storage. Third is low pressure and low oxygen storage. First is cryopreservation. Let's know about the cryopreservation in details. What do you mean by cryopreservation? Cryopreservation literally means preservation in the frozen state. The principle involved in cryopreservation is to bring the plant cell and tissue cultures to a zero metabolism or non-dividing state by reducing the temperature in the presence of cryoprotectants. Cryopreservation broadly means the storage of germplasm at very low temperature over solid carbon dioxide, that is at minus 79 degrees centigrade, low temperature deep freezer, that is at minus 80 degrees centigrade, in vapor phase nitrogen, that is at minus 150 degrees centigrade, in liquid nitrogen, that means at minus 196 degrees centigrade. Among these, most commonly used cryopreservation is by employing liquid nitrogen. At the temperature of liquid nitrogen minus 196 degrees centigrade, the cells stay in a complete inactive state and they thus can be conserved for long periods. In fact, cryopreservation has been successfully applied for germplasm conservation of a wide range of plant species like rice, wheat, peanuts, cassava, sugarcane, strawberry, coconut. Several plants can be regenerated from cells, meristems and embryos stored in cryopreservation. Now, let's know about the mechanism of cryopreservation. The technique of freeze preservation is based on the transfer of water present in the cells from a liquid to a solid state. Due to the presence of salt and organic molecules in the cells, the cell water requires much more lower temperature to freeze compared to the freezing point of the pure water. When stored at low temperature, the metabolic processes and biological deteriorations in the cells or tissues almost come to standstill. Now, let's know about the limitations of successful cryopreservations. First point is formation ice crystals inside the cells should be prevented as they cause injury to the organelles and cells. Second point is high intracellular concentration of solutes may also damage cells. Next point is sometimes certain solutes from the cell may leak out during freezing. Next point, protectants also affect the viability of cells. Next point, the physiological status of the plant material is also important. Now let's know about the technique of cryopreservation. The cryopreservation of plant cell culture followed by the regeneration of plants broadly involves some stages. First stage is development of sterile tissue cultures. Second stage is addition of cryoprotectants and pre-treatment. Third is freezing. Fourth is storage. Fifth is thawing. Sixth is reculture. 7 is measurement of survival or viability. 8th is plant regeneration. Now we will discuss details about these stages. First stage I told you about development of sterile tissue culture. Let's discuss. The selection of plant species and the tissues with particular reference to the morphological and physiological characters largely influence the ability of the explant to survive in cryopreservation. Any tissue from a plant can be used for cryopreservation. Example, meristems, embryos, endosperms, ovules and seeds. Cultured plant cells, protoplast, calyces. Among these meristematic cells, among these meristematic cells, suspension cells cultures in the late lag phase or log phase are most suitable. Now let's know about the addition of cryoprotectants and pretreatment. Cryoprotectants are the compounds that can prevent the damage caused to cells by freezing or thawing. Freezing point and supercooling point of water are reduced by the presence of cryoprotectants. As a result, the ice crystal formation is retarded during the process of cryopreservation. Thank you. We'll discuss next part in the next video.